The Claisen-Schmidt condensation reaction is an age-old reaction, first being reported by Claisen and Schmidt concurrently in 1881. It involves base-assisted enolate addition of a ketone or aldehyde, shown in blue, into an aromatic aldehyde, shown in red. Initially, the addition of the enolate results in the formation of an alcohol, as shown in the brackets. However, a subsequent dehydration ultimately produces an enone. Since the aryl aldehyde does not contain an alpha hydrogen, it cannot form an enolate. As a result, often weak bases such as hydroxide can be used to generate the enolate. The Claisen-Schmidt condensation has been used to generate a number of compounds over the years. However, it has been extensively used to join the aromatic ring systems of the chalcones and flavonones, which are shown in red and blue. The blue portion indicates that derived from the enolate, and the red is of the aromatic portion. The chalcones and flavonones are a central core to a range of biologically active molecules that possess a range of activities such as antibacterial, antifungal, anti-inflammatory, and anti-tumor, depending on the substitution pattern. Another broad class of molecules generated from the Claisen-Schmidt condensation reaction is the methine-bridged compounds, of which we give examples in this study. Our lab is interested in fluorescent constituents of the pigment bilirubin, which is naturally produced as a degradation product of heme. The synthesis of bilirubin and many of its constituents revolves around the Claisen-Schmidt type condensation, which can be visualized in the structures displayed by